Well, good afternoon. Thank you for the opportunity to be here and present my work. Well, not only my work, of course, the students. For many years, I'm dealing with the identification of mesenchymal stem cells, and I will tell you a little bit about their differentiation fate in health, in disease, and potential therapy. Well, the stem cells are known to build up the embryonic in all mammalian, and this is a very fast and fascinating process. And what I will talk about today is about the adult stem cells, those cells that are staying in our body from early birth until the last day of our life. Those cells that are basically dealing with normal remodeling and regeneration of all tissues, and we are talking about more than 200 kinds of cells, and we have to understand how they are functioning, since if something is going wrong, they will not perform well. And the main focus in my lab, will be about the mesenchymal cells. מה קורה עם העכבר? איזה כפתור? אוקיי. נכון. אבל זה לא עבד. So I'm talking about the adult stem cells, and I am focusing mainly on the mesenchymal cells. And I will try to show you how we can combine the different nanotechnology that are supporting the area of biological technique development and in medicine. And I will mainly focus on molecular studies, not too much molecular, about the various cells that we are dealing with and how we can develop them into medical devices. We have the understanding that this is a multidisciplinary area, and this is a synergistic in order to understand all these levels. So the stemness of mesenchymal stem cell is the focus. Well, the three main lineages that I will talk about are the cells that are differentiating into osteogenic differentiation, into the myogenic differentiation, and the adipogenic lineage. However, this is the normal pathways. If something goes wrong, we have the understanding, or we see in vivo, that if there will be less differentiation into osteogenic or myogenic, we will get more adipose tissue in our body. So bone and muscle are directly associated with adipose tissue, and there is interplay between the differentiation pathways. Adipose is very important tissue since it plays a major role in glucose storage, but when it goes wrong, it's also the one responsible for obesity. But the control of this mesenchymal cell being osteogenic or myogenic versus adipogenic is the major problem in pathophysiology. Sarcopenia is a condition where cells, instead of being myogenic, they will become more adipose, and this is in the muscle tissue. The muscle tissue will lose mass and will be atrophy of the tissue by adipose cells on the account of the myogenic cells. Very briefly, can be seen here that if we look on the a myofiber, we can identify the stem cell of the fiber, we can culture them in vitro, we can differentiate them into myogenic cell. However, under some conditions, we get cells that are not myogenic, they are fibroblastic or adipogenic. We have the same phenomena in the bone. In young animals, it happens also the same in human versus old animals, or in a model of ovarectomy means that we are losing the control of estrogenic hormones. We get in the bone marrow of cells more adipose cells. So the growth of intramedullary bone fat accumulation caused to increase in bone resorption and to the loss of bone structure, and this is osteopenia. So we are already seeing two kinds of pathologies which are related to the skeletal tissue, that we have more adipose cells on the account of the local cells that have to maintain the tissue. We have the understanding that there is an alteration in ECM that cause all these kind of changes. So definitely there is a need to understand why there is increase in fat accumulation, and this of course comprises the regeneration of these tissues, and the need to understand the stemness, how the cell decide which lineage fate to go is very important. Over the years we have done many in vivo and in vitro studies at the molecular level, genomic, metabolomics, transcriptomics, etc. And all these are nanoscale systems. So the cues for stem cell fate are multiple factors. We cannot rely just on one protein or one drug to make everything. We have to understand how the cells are sensing their niche, what are the factors, the secreted factors, cell-cell interaction, and physical factors that are affecting this activity. Maybe to find some algorithm that will tell us how cells are going into one specific direction, how the stem cell, which is a fibroblast-like, will decide if to go and make more of the skeletal tissues or alternatively into the adipose tissue. עכבר לא מגיב. לא צריך עכבר. כן, אוקיי, בסדר, הלכתי עם הדבר הזה. So the interplay between the different pathways is mostly important, and also we have to understand that the adiposity in this tissue are not only pathology of what we are doing here now, enjoying the food. It's not only nutritional effect, but it's also caused by style of life. Immobilization means there will be less skeletal tissue and more adipose tissue, as I showed you. So we can follow things maybe easier than just going into high molecular biology technique, looking by cell morphology and how they are changing and how they are translating their niche into the differentiation fate. This is an in vitro model to follow up the differentiation from the stem cell, from the fibroblast into adipose tissue, and we can see that there is a transition stage. The cell will differentiate under some conditions from the fibroblast into the adipocyte cells, okay? And they will change their morphology. They will become smaller. They will round up, and they are changing the ECM, and they are filling the niche. They are filling the stiffness of the niche, and they are filling the interaction between the different cells. There are changes in their cytoskeleton lead to different reaction and the different pathways of the final differentiation. Sorry. You can see how the readout and the interaction between cells relies on the change in the ECM component from less fibroblastic or fibrotic ECM to softer ECM, which is basement membrane-like, and the changes in the cytoskeleton. And this brings us also to the interaction between the cell. And the new approach is to look not only on secreted factor and the interaction of cell with the ECM, but also how they are sending natural nanoparticles in this community. So they are communicating by microvesicles or exosomes, and the communication is very important. What are these microvesicles or exosomes that are secreted between the cell, and how they are affecting their fate in differentiation? So we have to meet the natural nanoparticles at the cell niche in order to understand how the cells are functioning, and this can give us some ideas about potential new drug delivery system and potential drug therapy. What I'm showing you here is that if we take two kinds of cells, one was labeled with PKH26, means that labeling the cell surface, and then co-culturing with another cell. There is a transition of microvesicles between the two kinds of cells. It's very well shown and recognized, and this means that we can affect one cell by other cell in the same neighborhood. But what they are containing and how we control this. For example, the green cell population here that is cultured with the red cells. So the green are knocked down for some specific protein, and the red one are GLUT4 positive cell. GLUT4 is a very important transport for getting glucose into the cell, and it is essential in order the cell to become adipocyte and to gain their lipid droplets and the differentiation pathway. The knockdown cells will not be able to do so, but when they are co-cultured together, we can see that there is a transition of the GLUT4 positive microvesicles into the cell, and this accelerates also the other cell to become adipocyte. We can also isolate exosomes from conditioned medium of adipocyte cell. So by being able to understand what the cells are signaling and what kind of natural nanoparticles they are sending one each other, we can better understand how to control the cell niche. So we did mass spectrometry analysis to reveal the different kind of proteins that are expressed by exosome in this related system. And we have a list of proteins that I will not go through between fibroblast, undifferentiated stem cell, or adipocyte, and we see that they are expressing different proteins. And this is a clue how we can progress into developing of potentially drugs to control the system, as I showed you in the beginning. We don't want always the adipocyte to overtake cells in our skeletal muscle or in the bone, as it will affect the whole strength of the tissue. So we can develop biomimetic material, and we heard a fascinating lecture just recently by Nathan, how we can go for the different technologies. It was really amazing. So trying to work on the balance between skeletal cells and adipose cells has different approaches, and the chemistry here on ground can assist with developing such pathways and drugs. So the first part that I have shown you has some bad news on stem cell biology, that immobilization by lifestyle and static load or obesity or diabetes will increase the fat tissue not only in the adipose tissue but also in skeletal tissues on the account of the skeletal cells. And we have to learn how to maintain the cell niche, to study the cell activity, and this can be a platform to develop new drug for these systems. The second part of the talk will talk about other aspects of nanotechnology in developing devices and material used for tissue regeneration. Tissue regeneration in the skeletal tissue is very important in cases of orthopedic or dental, as we heard yesterday, if there is a hole that needs to be repaired, if there is any problem with tendons or other connective tissue, potentially building up artificial disc, or also maybe artificial blood vessels. So what is the potential or ultimate material for scaffolding, for scaffold material? We also heard about it just yesterday and today, and I will bring you some new approach. We have a scaffold made from non-mammalian material. What you see here is collagen fibers and also coming from soft coral. And this was first discovered at my husband's lab. He is a marine biologist. And using these collagen fibers, and we have shown that this is collagen by different technique, mass spectroscopy and NMR, and of course histological staining, can be built up biocomposite by making these collagen fibers together with hydrogel. And we have shown in collaboration with Professor Rami Khajali from mechanical engineering and the collaborative student that it will hold different potential for stress strain activity, depends on the density of the fibers and their orientation. And in vitro we have shown that the cells love this material. Cells are growing on the scaffold for many, many months, so there is no cytotoxicity, and they are forming in vitro tissue-like structure. And of course, this can be very easily translated into the in vivo. We have shown that cell can differentiate on this nanomaterial, and they can nicely absorb nanoparticles that can direct them to any direction that we would like in the tissue regeneration process. So in the last, or the second part, I have shown you that we have natural non-mammalian origin material, and this has a huge benefit since it doesn't have to go through chemical extraction, and it maintains a very well-defined mechanical properties. We can use them, these fibers, together with alginate, that the gelation is done under physiological condition in presence of calcium ions, and it's free of chemical or any other toxins. And the alginate embedding of collagen allow easy handling of this biocomposite, and cells are growing very well in these scaffolds. So we have multi-phase nanotechnology in order to assist in the biology and medicine, and I would like to thank your attention, to thank the agencies supporting the studies, the current student in the lab, and you for your attention. Thank you.
Thank you for a wonderful talk. Okay, we already have questions. Ben?
Terrific talk and very interesting results. I have a quick question regarding the first part about the role of the microenvironment on the differentiation of these cells. Did you look at the different aspects of this?
Of course.
And
What specific questions you have?
That,
You may get another
yeah.
2 hours lecture, yes.
yeah, so the ECM obviously will have a significant role, and the ECM also might change in different conditions of the development.
Yes, definitely.
So how this might affect as well on the differentiation of the adipocytes and the more fatty layer that you have?
Well, as I mentioned, ECM is different in its consistent as well as in the stiffness, will affect the cell mechanobiology and the pathway that they will go. This is in short.
Hi. My question is, have you looked at the immuno response to the coral collagen in vivo models?
Well, this is a great question. Of course, we are now doing the in vivo studies, and this biocomposite were implanted in mice for rat for a few months, and there were no immune response problems.
How much the coral tubes can survive in normal physiological condition? I mean, I'm guessing that the sea is rather salty, right? So does the regular low salt environment, like in our physiological conditions, is good enough for them to sustain their structure?
Well, salt is not a problem. Potentially problem can be proteolytic activities, but as I showed, cells are living on this scaffold and maintained for several months, and there is no problem in their stability.
Okay. Cool. So let's thank Dafna again.
Thank you.